So we've talked about viral unit structures. They're not cells. Unit structures, viron, bacteriophage, and we talked about the life cycle of a virus, including the two different types of life cycles of bacteriophage. I'm going to talk just a little bit about bacterial or viral infections. And when we talk about viral infections, because viruses are ultra microscopic, we can't look at a virus and diagnose an infection. So we use indirect means, and mostly we look at the damage that it's done to the cell. And that's called the cytopathic effect, the viral-induced damage that can be seen microscopically. And you don't have to know these specific names, but I just want to give you a couple examples. So inclusion bodies, which are masses of virons, viruses, inside the cell, or damaged organelles, depending on where they're replicating, are things that we can see. We can also see syncytia, which is fused host cells. So sometimes when the host cell machinery gets hijacked, it does weird things, and it starts making multiple nuclei. Viruses are kind of similar to cancer in that they force a cell to do transcription, translation, and replication unchecked. And so if you remove those checks from DNA replication especially, then sometimes the host cell starts replicating DNA unchecked. And so you start seeing multiple nuclei in a sample. So here's an example of that. Multiple nuclei in one cell, that's not good. That is a cytopathic effect. And then this one would be also multiple nuclei. And here we can see inclusion bodies. You can see a huge inclusion body that there's something going on there. It shouldn't be that shape. And it's probably because it's full of viral particles that we can't see. So the results of viral infections, now these viruses, if they don't leave the cell, it's called a persistent or chronic infection. For example, measles can live in the brain for years. Some viruses can go latent. What happened? There we go. Some viruses can go latent for years at a time, and if they do that, you don't see signs and symptoms because they're not replicating, but they are still in the host. And so that would be things like herpes, which can have flare-ups, right? So like herpes simplex, which causes cold sores, or genital herpes, which comes and goes. You always have herpes, even if you don't have the cold sore or the genital warts. Chickenpox can remain latent for decades, and when it goes back active, it manifests as shingles. So that's an example of latent viruses. Now, there are viruses called oncoviruses. If I'm telling you oncoviruses, what does that make you think of? Cancer. Yep. And I mentioned that already, right? So some viruses can permanently alter the DNA of the host and cause cancer. That means that the cell just replicates unchecked. A lot of times it's larger, it's faster, meaning replicates when it's not supposed to, and faster, so it doesn't complete the cycles properly. The membranes aren't complete, chromosomes aren't complete, it's just a mess. And some viruses can cause cancer, and actually in my lifetime, human papillomavirus, HPV, was determined to cause cancer, and they came out with a vaccine for that. There are about 9 strains of it. And so there was a threshold age, it was early 20s, that girls especially were supposed to get this vaccine before they had been exposed to any of the 9 different strains of HPV. So let's talk just a little bit more about viruses, culturing viruses. Now, when you culture a bacterium or a yeast or even an alga, you put it on a medium and you give it food and it grows. It replicates, it does its metabolism. But remember, viruses don't have their own metabolism. So they necessarily need to have a host cell. You have to give them a host cell to do the replication for them. And so growth has to occur in the host cell, and it has to be the correct type, either bacterial or animal or plant. And you can even use things like bird embryos, and that's why when we are replicating virus for vaccines, a lot of times the warnings say don't get this vaccine if you're allergic to egg yolks, because that's how they actually replicated the virus. Some animals are used, but and I champion this only if it's really, really necessary. There are a lot of rules and regulations for what you can do with an animal that has a vertebra. And using a vertebrate for science requires a lot of regulations and paperwork, and so it's only if it's absolutely necessary. Now, when we are replicating them in culture, in bacteria or just a tissue culture, like human cells, but in a lab, the way we see a virus is we see them killing the cells. So that's called a plaque. And that's the area of cell death. So with a bacterium or, say, a yeast, you see the cells. You see a colony, right? You see them grow up. Instead, so let's look at this one here on the left. This is a bacteriophage. This is agar that has been mixed with living, say, E. coli. And so the agar itself is turbid with living cells of E. coli. So the E. coli become the media, really, and the virus invades the E. coli cell, and the E. coli cell replicates it, and then it lyses and explodes, and then that virus goes to the next cell and replicates and explodes, and so on. And so you start to see these like little pinpricks in the opaque agar, in the uniform turbid agar there. And they get bigger and bigger over time because the virus continues to replicate as long as there's a living host cell. They start out as just tiny little pinpricks and they get bigger and bigger. But these clearings are the plaques. And then over here, it's the same thing, only this is a human tissue culture. So human cells are completely clear and colorless, and they don't form colonies. It's actually important that human cells form a single layer. If they start to pile up on each other, that's a problem. That's a cancerous type cell. And so they form a flat, thin layer on the bottom. This is like a well. It's like a little cup, a little plastic cup, and it's formed on the bottom of that. And you can't see human cells. It's just a very thin, clear, colorless layer. And so they've stained it. They've stained it probably crystal violet or something to make the human cells colored so that we can see where there is no cells. And this is probably a titer, a way of measuring the number of viral particles. And so you can see they did a serial dilution here. There's no virus, one plaque. So there was one viron in that original titer, that original serial dilution, and then a few more here, and then more here, and more here, and more here, and so many in this particular well that it just killed all the cells. There were enough viral particles to kill all the cells in one shot. They didn't need to leave one host cell and go to the next. Okay, so that's how we culture viruses. We talked a little bit how we can see viruses. But in the lab, how do we determine if somebody has a virus? Yes, we look at cytopathic changes. We might use immunofluorescence, so we might take antibodies that are specific to, say, like viral proteins or cell molecules that a infected cell would make, and they would be tagged with a little fluorescent tag. So when they connect with your tissue sample or your viral sample, they would glow, and you would use a special light to see where that virus is then, or if it's there. You could use electron microscopy, but that's really impractical and unlikely. Most commonly what we use is something called PCR, and the way this works is we can actually make DNA in a test tube. We can take all the ingredients, all the nucleic acids and the enzymes and the things, the buffers and things that we need, and we can mix them in a test tube. But what we do is we don't give it a template. We only give it the start and the end of the recipe, and if the template is there, if the viral nucleic acid is there, then we can make the DNA. So then when we check our PCR to see if it worked, we look, did we make DNA? Oh, if we did, the virus was there because the template was there. If we didn't make any DNA, then there was no template, there was no virus. And again, we could detect antibodies. This is a very indirect assay. This is kind of one of those rapid assays where we would use like rabbit or mouse serum, where they've been exposed to this virus and they've made antibodies, and we could connect to see if our body's making those same antibodies. Treating a virus. Okay, so this is really difficult. Okay, think about just adsorption. Right, the receptors that are part of the host cell that are necessary for viral adsorption aren't there hanging out going, well, just in case a virus is here, I'll just have a receptor to accidentally bring it in and get infected. No, those receptors are there for cell-cell communication, for host cell communication. And so they're necessary for normal host function. And so we can't give somebody a drug that's going to prevent adsorption, because that's going to disrupt normal host cell function. Same thing with the machinery, right? The machinery of the host is necessary for normal host cell function. We need to always be replicating our DNA, transcribing, translating. So if we stop that, we're going to kill our own cells. It's very difficult to treat a virus. Our best bet is to vaccinate before, and even that's limited, and we're actually going to dive into Unit 4. We're going to design our own vaccines. So we're going to have a little taste of what it's like to try to design a vaccine. Oh, as if you forgot that there would be some weirdos, right? There's always weirdos. So this topic is called Introduction to Viruses, but the truth is, viruses are acellular infectious agents, right? Infectious agents that aren't made up of cells, but there are other acellular infectious agents, such as prions, right? So prions are also acellular infectious agents, and what are prions? They're not nucleic acid and protein, they are just protein. Proteinaceous infection, prion, or infectious protein, if you want to say it that way. I still have a hard time spelling proteinaceous. Proteinaceous infection, yes. So these guys are thought to be normally folded proteins that have been misfolded. So these are proteins that our body makes, but they've been folded wrong. And the way that they replicate, I'm doing air quotes here, the way they replicate is that when they contact the normal folded protein, they misfold them. And so that's how they perpetuate. And because our body isn't designed with the enzymes to break down these misfolded proteins, and they don't interact with normal host cell components properly, they start to accumulate, and they usually accumulate in the brain, and that's going to cause massive, massive damage. Can you think of prion infections? Most students think of mad cow disease, bovine spongiform encephalopathy. spongiform meaning brain sponginess, right? So mad cow disease. There's also Creutzfeldt-Jacobs disease, and also chronic wasting disease, which is very significant in Michigan because the white-tailed deer population now has this prion. And these things are extremely resistant to disinfection because they're not alive, and even some sterilization. So proteins are three-dimensional structures, and most of the time we can destroy a protein by unfolding it. That's called denaturing it. And we can do that by heat or acid to break the bonds of that three-dimensional structure, tertiary structure. But once you remove that heat or acid, they can just refold or renature. So that means that even if you killed a white-tailed deer that had this prion disease and you cook the meat quite thoroughly, you are still at risk. When I was younger, mad cow disease was rampant for a small stretch, and it was really tragic because they had to kill all the cows, and they couldn't do anything with the meat. They had to just incinerate them. They were just burning cow carcasses out in the fields. It's really, really tragic. So there's one more infectious agent that is acellular. It's called a viroid, and this is a real weirdo. This guy depends on viruses for replication. It is just naked nucleic acid, and it kind of supports the idea that maybe viruses came from just naked nucleic acid and somehow coded for a protein coating, because remember, we still don't know where viruses came from. Viroids we don't hear about much in human pathology, but with plants, it's especially important. And this is always a fun little story that I like to end on. So there was something called Dutch tulip mania, and it was back in the 1600s, where in Holland there was kind of like a stock market on tulip bulbs, where people would bid, like auction tulip bulbs, but they would auction like future tulip bulbs. So like not even ones that they actually had. Like you didn't have the actual tulip bulbs. It was like, well, I have this basket of tulip bulbs, and after I plant them, I'm going to have 100 baskets of tulip bulbs, and I'll sell those to you. And these became very prestigious. Like people would display their baskets. They would they would mortgage their farms. They would sell their houses so that they could invest in these bulbs of tulips, on the promise that once they were planted, they could then get them. And what happened was the price of tulip bulbs just skyrocketed. And then at some point, somebody said, "Hey, wait a minute, we're mortgaging our farms for a promise, and I'm not paying that much for a promise." And so then all of a sudden, the worth of those potential tulip bulbs just crashed, and people lost all kinds of money. They lost their life savings, their homes, their farms. And when they were when they were auctioning these tulips, because remember, they weren't real flowers at the point, they were using cards, pictures of what that tulip would be. And these variegated tulips with these really pretty striping, were the most desirable tulips. Those were the ones that went for the most money. And the reason that those tulips are variegated is because that plant is infected with a viroid. So it's kind of interesting that the ones that were most desirable were the ones that were actually the most sick. Okay. Couple roundup questions here. The general steps of viral multiplication. Yes, it's A. What's missing? Uncoding. A prophage is a blank stage in the cycle of what? Latent, right? And if it's a prophage, what life cycle is that? Lytic or lysogenic? Lysogenic. Okay, in general, an RNA virus is going to multiply in the cell blank, and a DNA virus is going to multiply in the cell remember, DNA to RNA to protein. DNA starts in the nucleus, RNA replicates or is translated in the cytoplasm. Viruses that are persistent in the cell and cause reoccurring disease are called latent. Structures of a virus. Okay. Which of the following is not a pattern of viral morphology? You can have enveloped or non-enveloped, so you can have enveloped or naked. It could be helical or icosahedral or complex, but you wouldn't have complex and helical. Right? Complex means it uses fibrils instead of capsomers. Okay, NCLEX prep question. Oh, so they love these select all that apply. So the way that you should treat these, and this is part of the why I design my questions the way I do, is treat each one as a true or false question. Okay? Okay, I'm going to switch it, so if you need to pause it for more time, do it. Viruses do not have a cell. They are not active outside a host cell. All the rest of these things are true. Got it? Can be present in latent state. Yes. Alright, here's another select all that apply. Okay, I'm going to switch it, so if you need to pause it. So, nausea and vomiting can happen with a virus or a bacterium. Blood cultures, if you can culture it, in that case, yes, you would be able to tell the difference. You could also use blood for like that PCR, that template test that I told you about. If it's a bacterium, you can do rapid strep testing. You would absolutely not want to give somebody antibiotics and see if they got better. That is a gross misuse of antibiotics. And that's part of the reason why we are running out of antibiotics for use for treating sicknesses. CBC, I guess I should have explain that. That's a complete blood count with a white blood cell differential. So that would work too, because oftentimes if a virus is causing a white blood cell count to increase, or if we have an increase of say, our phagocytes that would be responding to a bacterial infection, we would be able to tell the difference between a viral and a bacterial infection. Okay, I think this is the last one. Okay, I'm switching up. It is all of the above. Now, I know as a microbiologist, I have really emphasized that this word means nothing living. It actually means no living contaminants. So it is still a sterile environment. Think about like your heart is a sterile environment, right? Your lungs, your eye is a sterile environment. Definitely important for metabolism and replication, and they are self-supporting, so that's part of why we use them. Okay, so this is the last one. So I don't test on examples, but NCLEX clearly wants you to have a little bit of a repertoire, and I did mention these. I mentioned the human papillomavirus, and what does it cause? It causes cervical cancer. So papilloma is not going to cause brain cancer. Herpes doesn't lead to AIDS. What leads to AIDS? Human immunodeficiency virus, HIV, leads to AIDS. Hepatitis B does cause liver cancer. Yes, it does. Probably because it replicates in the liver cells, right? And then there's papilloma again, but not gastric, cervical cancer. So it's hepatitis B. Okay, that's the end of this video too.